right. Hello, everyone. Welcome to part three, section three of our MCAT strategy series. If you've joined us for the first two, great. If you haven't, definitely check out the recordings because we've dedicated one per section over the last month or so. So I'm excited that you're here. Go ahead and say hello in the chat that says introduce yourself. Would love to know where everybody is dialing in from and what you're most excited about today, I guess, is what it comes down to. My name is Petro Spinazzi. I'm the VP of Test Prep and Tutoring here at Med School Headquarters. I've been involved in the MCAT space as in multiple different capacities for the last 25 plus years or so, actually more than that now. And, teaching, designing curriculum right now, heading up our test prep and tutoring services here in Med School Headquarters. So we are really a one-stop shop in terms of getting you into medical school, whether it's MCAT prep, advising services, all the things. We can help you with that. And so we're super excited to have you here tonight. Specifically, our focus is the biochem section. I will not call it the Biological and Biochemical Foundations of Living Systems section, aside from that one time, because that is a mouthful. So we'll just call it the biochem or the bio section to make life even more efficient. So that's what we're going to be talking about here tonight in terms of not just the section, but kind of what it really is ultimately testing you on. And then more importantly, kind of how it fits into the entirety of the exam. And we'll take a look at an example passage along with a couple of questions so you get a sense of kind of how you'll be tested on this exam in this section very specifically. Today, our focus is going to be on the biochem section. However, the passage and more specifically the questions that we're going to look at are a very specific type of question, or there's some examples of very specific types of questions which they ask you to incorporate new information in the question itself. So the question will actually add in something and say, what if we did this, right? What if this happened? And that's a very common question on the MCAT. We haven't really looked at those in the first two sessions, so I thought it would be the right thing to look at here today. Even though we're looking at this in the context of the bio section, those do show up in the other sections. So don't think that this is exclusive to this, but I figured as a means, kind of give you a good breath, throughout the course of the entire series, give you a good breath into exposure of the different types of questions. That would be a good one to kind of look at and focus on. So we'll talk about that as things progress here. This session is ultimately designed for you. So if you've got questions about things along the way, feel free to ask them. If you haven't already registered for MAPTCon 2025. It's on September 13th. It's completely free. We're going to have all sorts of different sessions specifically for pre-meds throughout the day. And at the same time, lots of time with various medical schools and medical programs that might be of interest to you. So if you want to get to know more about those programs or get to know the people that are behind those programs, MAPTCon is a great event for you to be part of. Completely free. Come and go as you wish throughout the course of the day. I'm super excited about it. It's only 2 weeks away or so. I know Emily, who just posted the link for it, is super excited about it because she is the driving force behind MAPTCon coming together and is doing an awesome job. And really, we are really looking forward to welcoming each and every one of you to that event and hopefully getting you some great information, some great exposure as it were. So please come. And there'll even be an MCAT session that day. So, you can get more MCAT fun is what it comes down to. That being said, let's dive into kind of overall MCAT progression. If you've been to one of these before, any event that I do, I always start with this slide for a very specific reason, because we always want to remind ourselves that really studying for the MCAT, it's a long process, right? This is not a process that happens overnight. This is not an exam that you can cram for in any way. So it's really, really easy to think, oh, I'll just cram for this exam like I would cram for any other exam. You can't. It's impossible. When you are preparing, you will be adapting along the way, and your preparation will never really ever be the same from day to day. namely because earlier on in your preparation, that kind of dark blue triangle that's there in these conjoined triangles, that's where you're going to be spending time as far as content review and actually developing your critical thinking skills. In the early days, off to the left, there's going to be more of that. And that is going to diminish over time because you're going to be incorporating more and more application and practice in the development of your endurance skills. So it's really an evolving process. And that's, I think, a mistake that a lot of students make is they think, oh, I'm just going to do all this content review and then I'm going to work on questions. No, you do some content review and you start slowly incorporating questions until you get to the point that you're taking full-length exams. So it is a progression of time that is important for you to recognize and to kind of be mindful of as you're working through that. If you need more specific guidance or want more specific guidance, that's of course where our tutoring services can definitely help you out and helping you kind of build out these study plans and a study plan that's very specific to what your needs are as you're going through. So by all means, reach out to us and we can help you along the way for that as well. So when we're looking at the MCAT prep progression, we have to also be mindful of the fact that you do have 4 different sections and you have lots and lots of different topics that you're going to be responsible for, and is pretty much fair game on this exam. When we look at the bio section specifically, we want to make sure that we are in the parameters of what can be tested in the bio section. But more importantly, recognizing the fact that the sciences are integrated with each other. So you may see questions that are more, physical science related, even in the bio section, which, throws students off sometimes. But the reality is that is very, very, very possible to happen. The structure of the exam as a whole, I won't dive into this because you've seen this 100 times before. If you haven't, it is all over the world, so it's impossible to miss it. But the structure of the exam, very straightforward. It's 4 sections. It's a long exam, right? It's a very, very long exam. But the key to recognize is that the 3 sections that are science-based all have the word biology in them. And that's not just titles. That is fair game. Everything is in this biological context. Even in the chemical and physical foundation section, you are being tested on those topics in the context of the biological sciences, the biological systems. When you get to the biological and biochemical foundation section, they couldn't say biological and biochemical foundations of biological systems, so they said living systems. But the reality is that you might see some physics in there. You might see some chemistry in there. And, very easy to kind of miss the mark there and realize, there, these other sciences, they can show up because it is all interconnected in very, very, very specific ways. So when we look at the biochem section, as far as the content that is fair game, there is biochemistry. About 25% or so of the content that you'll be responsible for is going to be biochemistry. If you go back and look at, my slides for for the chem phys section, you'll see that it also says that there's about 25% biochemistry. Biochemistry actually shows up pretty equally in these 2 sections. In terms of introductory biology, of course, that's going to be there. But remember, introductory biology has elements, very much so, of physics and chemistry that's built into it. So you'll see some general chemistry. The misleading one on this page is the organic chemistry. You think, it's only 5% of this, this section of this test. I don't really need to worry about it. The reason why it's misleading is very simple. Organic chemistry is the foundation for the bulk of biochemistry. So you need to know your OChem in order to know your biochemistry. So while it's not explicitly tested as much, it is implicitly being tested in the biochemistry portion of this section. So just something to bear in mind as you're looking through this. Don't be misguided into thinking, I don't need to worry about OChem or Orgo, depending on what abbreviation you use. That subject is very much going to be there, but it's going to be showing up in the other sciences or in the other, in the biochemistry and in the biology as you're going through. So something to kind of remind yourself of as you're studying that material. It is important. So I'm sure my OChem professor will be happy to know that I just said that OChem is important, because he was very adamant that OChem was important. The MCAT, any question on the MCAT is going to be at the intersection of content and one of these skills. So the 4 core skills are knowledge of scientific principles, scientific reasoning and problem solving, reasoning about the design and the execution of research, and then database statistical reasoning. So when you're looking at a question, you're being asked to do multiple things. You're being asked to recall content. You're being asked to demonstrate one of these skills, and then at the same time, you are being asked to read a passage and extract information from there as well. So there's a lot going on in any one given passage. Sometimes, in fact, you'll have kind of multiple skills that may be tested, or it's really kind of your job to figure out which skill it is that they're testing you on. The design and execution of the research, that's an interesting one, because there you're not being asked to design anything. You're being asked, why did they do this, right? Why was this a good choice, or why was this a bad choice, right? What mistake did they make in deciding in designing this experiment? So that's something that will come up, and it's a little bit about really understanding how experiments are set up without actually having to do the experiment or do the research, as it were. As far as studying for the biochem section, I mean, you can write this down for any of the sections. I'm going to be perfectly transparent. I didn't change anything if you came to the Chem Phys one, because it's the exact same thing. You need to be relearning the fundamentals, right? It may have been some time since you've seen this material. So you need to make sure that you are learning or relearning those fundamentals. You have to make sure that you are practicing what you have learned. And then you have to always, always, always revisit what you've learned, right? It's not good enough for you to say, okay, I've learned this, now I'm done with that, I'm going to move on. You have to always be revisiting this information, which really brings us to this concept of interleaving. Now, I chose some very specific letters for the purposes of today. I could have very well easily have just chosen A, B, C, and D, but I thought it'd be fun to choose A, T, C, and G, because, of course, DNA, and we're talking about bio. But when you're thinking about the concept of interleaving, the concept of interleaving is that you are constantly kind of mixing things up, right? The last thing you want to be doing is studying all of A, right? All of this, and then moving on to studying all of T, and then moving on to studying all of C, and then moving on to studying all of G. Because what's going to happen by the time you've studied all of G, you will have forgotten the majority of the information about A, right? Whatever A might be. And this is, again, just a silly example for you. So the idea behind interleaving is that you are kind of constantly mixing things up and you're going back and forth. So maybe you do a little bit of, oxidative phosphorylation. And then you do a little bit of, let's call it genetics. And then you do a little bit of, throw in some DNA translation. And then you do a little bit of organelle structure. And then you go back and you do a little bit of oxidative phosphorylation. And then you can do a little bit, right? You're constantly kind of going back to the information from before. But as you're doing so, you're progressing along the way. There's a great article. It's from Scientific American from a couple of years ago, but it's still very, very relevant. I'd encourage you to take a look at it. It's called The Interleaving Effect. So it's very well done. It's a very quick read. It's like a 5-minute read or so, but it'll still give you a good sense of kind of how you want to be thinking about your MCAT prep. Really any prep for that matter. When you get to med school, you'll benefit from this as well. It's effectively it's the kind of most proven way as far as success for retaining information. So I would encourage you to take a look at this in this article. It does a really nice, clean job of explaining it. So that's why I always encourage students to take a look at this if they haven't seen this concept or want kind of a more in-depth explanation of how the concept ultimately is going to work. Okay, so take a look at that. That's something for you, kind of bit of homework, as it were. Plus, it will help you develop your critical reading skills and reading skills as you're going through as well. If you have questions, feel free to put them into the question chat. Victoria asked a great question. Are there any specific study programs that we recommend? And the answer is yes. We recommend medical school headquarters study programs. So, that's part of why I'm here. So we now offer full MCAT test prep services in terms right now in terms of tutoring, and coming soon to a theater near you, something a little bit more exciting in terms of ways to study and ways to prep for the MCAT. Stay tuned for that. That is coming up, as it were. So that being said, I thought it would be good for us to look at a passage. The best way to learn any of this stuff is to be ultimately kind of getting hands-on and getting dirty and things. So here's the thing. With the bio section, sometimes passages are very, very intricate, right? They're extremely intricate in terms of the information that's being given to you. And sometimes from a textbook, as it were. This one is that second category, where this could have very easily just come from a textbook. And you might read this and go, oh, this looks a little bit, looks a little bit easy, kind of. But that's always, always, always going to mean that if you've got a passage that might seem a little easier, that's going to mean that the questions are going to be more complex. Okay, so that's kind of the first strategy, the first lesson you want to learn as far as tonight is concerned, that it's not just about what you're seeing on the page here. It's about really what's going to kind of be behind the scenes and what it is they're going to be testing you on later down the line. So you'll see what I mean by this here in a second. So as you're reading through these passages, the key is going to be to not get lost in the details, right? And I say that to you because it's really, really easy. And if anyone's studied for the MCAT or has taken the MCAT, maybe you've had this experience where you find yourself just like getting overly stuck in trying to remember all the details that are there. And so generally speaking, what you want to be doing is as you're reading through these passages, you want to be identifying what is the reason that the author put, and I'll use an arcane term here, pen to paper, right? This idea of like, why did they write this down? I don't think anybody actually puts anything pen to paper anymore, right? When they write things, they put fingers to keyboards, or not even that, right? They just might speak out what they want to. At some point, we're just going to be able to plug into our brains and boom, into the computer goes all the information. So here's what we're looking at, though. We're looking at a passage that's going to give us a lot of information, right? It says DNA replication in human cells begins at thousands of origins distributed along each chromosome. Now I'm going to pause there for a second. Do we know that from before today? Like before you showed up at med school headquarters, MCAT strategies, biochem section, did you know the DNA replication begins at thousands of points of origin along a chromosome in human cells? Yes or no? You tell me. Tell me in either of the two chats. I think let's use the introduce yourself chat for stuff like this and keep your questions in the ask a question chat. Yes or no? Did you know this before today? I'm assuming you're going to say, yeah, Petrus, I totally knew this, right? It's not new information is what it is. They then tell us that helicase first unwinds the parental duplex. You say, yep, I knew that too, right? I remember reading that. The good news is if you forgot that it was helicase that first unwinds the parental duplex, they just told you that it was helicase that first unwinds the parental duplex, creating a replication fork in which single-stranded binding proteins stabilize the separated strands. You say, yep, I've seen that before. But if you forgot it, they just told it to you. Primase then lays down short RNA primers that provide the free 3' OH required by DNA polymerase alpha to initiate extension. Great. You're giving me more information. Polymerase delta subsequently takes over elongation on the leading strand, which, while discontinuous synthesis on the lagging strand, proceeds as a series of 1 to 2 kilobase Okazaki fragments. Do we know this information from before? Absolutely. Or more likely than not, right? We've seen this before. Here's my point, right? I'm going to make an emphatic point. I'm trying to be a little sarcastic, but not too sarcastic when I read this to you. This isn't necessarily new. You might be looking and saying, yep, I know this, But what that also means is, since they just told it to you, they're not going to reward you just for knowing it, right? So immediately you see a passage like this and you say, okay, they're giving me information. Now all of a sudden, the game that you're going to have to play against the MCAT is going to be a little different. The game you're now going to have to play against the MCAT is going to be a game in which you have to show that not only do you know this, because everybody knows it because you've read the passage, but do you understand what it means for you, right? Do you understand what it means if I start to throw some weird variables into the mix, right? So that's what we have here. The purpose of this first paragraph, quite frankly, pretty straightforward, is to tell us about how DNA replication gets initiated, right? How it all starts. That's what they're telling us, plain and simple, right? In order to get things going, we're going to need these RNA primers. They're going to need to, do these things, and that's what we're going to have happening. They tell us some terms in terms of the different polymerases that are there, but that's it, right? It's not that exciting of an opening paragraph, and that's fine, right? We're totally, anytime it's not too exciting, be like, all right, it's not too exciting now. It's about to get exciting later on. So that's what we have with that one. The second paragraph, and this one's because helicase unwinds DNA in one direction, leading strand synthesis is continuous, whereas the lagging strand must repeatedly reinitiate. We think, okay, well, we got our replication fork, right? I'll use my hands in the example. It's kind of opening up, as it were. So the leading strand, it just kind of keeps going. The lagging strand has to continually get started again. Each reinitiation requires a new RNA primer and eventual sealing by DNA ligase. So let me ask you this. What is DNA ligase in terms of, from a biological perspective? What's another way of thinking, or have you thought about DNA ligase? What would it be the equivalent of in our kind of usual day-to-day world? Maybe something that you used quite a bit when you were a kid in grade school, right? Or even in kindergarten or before in preschool, maybe. I don't know when we start using glue, but it's glue, right? It's molecular glue is what ligase effectively is, right? It kind of sticks everything together. They say that topoisomerase relieves the positive supercoils that build up ahead of the fork, preventing torsional strain from halting the helicase progression, right? So they're kind of giving us the different players. They're saying, here's one player, here's what the other player does, here's what everybody's doing as far as this process is concerned. If you think about replication forks like two shoelaces that have been tied, twisted around each other, and then you try to pull them apart, the shoelace gets the knot gets very, very tight all of a sudden. So it's a very similar type of situation that's happening. That's that supercoil that starts to build up and makes things more difficult to separate. So this is what we have going on, right? Second paragraph. Basically, they're talking about topoisomerase and helicase. Really, they're talking about helicase and topoisomerase, right? That's it. There's nothing more than that as far as the second paragraph is concerned. You get to the third paragraph, and you say, well, I hope this is going to get more interesting, because so far, they've given me background information. But that's fine, right? It's okay to just get some background information every now and then. They say investigators labeling newly synthesized DNA with a fluorescent nucleotide analog observed a constant incorporation rate of approximately 100 nucleotides per second. So we'll call that NT per S. So nucleotides per second on the leading strand. On the lagging strand, incorporation occurred in bursts of roughly 1,000 nucleotides every 10 seconds. So here's my question to you. If I have 100 nucleotides per second versus 1,000 nucleotides every 10 seconds, is that the same rate at the end of the day? Are they telling me the same thing, just in a different way because it's the MCAT? Absolutely, right? The MCAT they absolutely love to kind of throw you off because you're like, oh, this one was 100 nucleotides per second. The other one's 1,000. But they very subtly throw in there every 10 seconds. And you go, wait a minute, 1,000 divided by 10, that's 100. It's the same exact thing. Why didn't you just say it's the exact same thing? Okay. The difference is, of course, one is happening in bursts of about 1,000 nucleotides every 10 seconds, whereas the other one is more continuous, right? It's a continuous 100 nucleotides per second, because they say it's a constant incorporation rate versus these bursts of 1,000 nucleotides. But in the end, the overall average rate is the same, because it's the exact same thing, just going in different directions. Despite this start and stop pattern, the average overall rate on both strands was comparable. So they then tell you that. They're like, okay, if you didn't do the math, we're going to tell you that. When cells were treated with a topoisomerase inhibitor, fork progression slowed by 60%. But adding the inhibitor after unwinding had already begun, produced only a 15% decrease, suggesting the topoisomerase activity is more critical early in fork establishment. So they say, all right, here's our little experiment. We're throwing in this inhibitor of topoisomerase, fork progression slowing by 60%, but we put it in after we unwound things. It's only going to decrease it by 15%. So it's not as critical later on. It's more critical in the early stages of things. So really here, they're talking about this inhibitor that's ultimately being introduced. And that's the entirety of the passage. I mean, that's it, right? It's just like, here's what we found, right? If we did this, we found that. So then you scratch your head and you say, okay, what questions could they possibly be asking me about this passage? Because any question they ask me is probably going to be pretty, straightforward, as it were. And this is where the MCAT kind of comes into its stride, right? It comes into its stride because it's now able to say, you know what? Even though I've given you a relatively easy passage, which normally would only lead to easy questions, I'm going to try my best to try to throw you off. And this is what I was saying earlier, right? I'm not making this a us against the exam situation, but I sort of am, right? So the question you have to say is, all right, I need to be on guard because they're about to throw something at me that is going to kind of shake things up, right? So you get to the first question, and it says, according to the passage, anytime, write this down, don't forget this, anytime you see the words, according to the passage, that means you are going to need to go look at the passage in order to be able to verify your correct answer. Because they are literally telling you, look, the passage told us something. What was it? Right? So according to the passage, which enzyme synthesizes the RNA primers required to initiate DNA replication? So we need to initiate DNA replication, right? We got to get things started. So really, the question behind the question, and this is another kind of tactic that you want to start thinking about and start kind of putting yourself in the mindset of, you have to ask yourself, what's the real question behind the question? And the question behind the question here, very simply put, is, do you know what these 4 answer choices do? And if you know what these 4 answer choices do, then you'll be able to choose the correct answer. So even though you're looking for the one that is required to initiate DNA replication, really you need to know what all of them do so that you can find the one and choose the one that initiates DNA replication. Does that make sense? Right? This idea of like, okay, I need to know one thing in order to be able to solve the other. So I'm going to let's go through these. DNA polymerase delta, right? The passage very explicitly told us what DNA polymerase delta does. What does DNA polymerase delta do? What is its job as far as this process is concerned? Or don't even think about DNA polymerase delta, just DNA polymerase. The word polymerase means that it is the thing that is going to be making, right? It's going to be the thing that's going to be making the actual DNA, right? It's the guy, it's the one who's making it. So we know that information, right? So we say, okay, DNA polymerase delta, that's the one that, first paragraph said subsequently takes over elongation on the leading strand. So that's not the thing that's going to be initiating the DNA replication. DNA ligase, that's the glue. It's sticking everything together, right? It's going to stick together the Okazaki fragments. That's not initiating the DNA replication. Primase, it's the primer, right? It's the thing that literally, it says primase lays down short RNA primers that provide a free 3' OH group that's required by the DNA polymerase in order to stick on. I did some painting recently, and before you put your paint on, you got to put the primer on, right? The primer is stickier, literally, right? It sticks better to the old paint that's already on the walls, and it sticks better to the new paint that you're putting onto the primer. So it's nice and sticky, kind of primes things, gets things nice and ready for you. So same kind of concept there. So that's where the primase is going to be the one. Topoisomerase, of course, we read in the passage that it's the one relieving the supercoils. So the only one that's going to work is going to be answer choice C, right? And that's the extent of the question. This is pure recall recognition, right? Here's the information that we have. Here's the information as we ultimately see it. Okay, so correct answer on this one has to be C. But here's my question to you. Was that at all challenging? Yes or no? I know I walked you through the information, but what would you say? Like, you look at that, you go, overburdened, underburdened? Not really, right, Victoria? Yeah, it's not. Guess what? The MCAT every now and then gives you a gift of a question like that. So you know what you do? You accept the gift, you thank the MCAT, and you say, great, I'll take more of those if you have them, right? So if you get those questions, those are kind of, that's what it is. Now, Victoria, we did not plan this, right? But you very correctly are saying, but now you're scared for what's about to come, right? So you're like, okay, that's the first question. That's like the straightforward one. Now what's about to come? And I don't want to overkill it. It's not going to be impossible, but they've got to give you something more of a challenge, because they're not going to give you, let's call it 5-6 questions that are as straightforward as this one, right? Every now and then there's this is basically a reading comprehension question in a science passage, right? Can you go back to the text and find me the thing that says it's required in order to initiate the DNA replication, or rely upon your content knowledge to show me that? So yes, that's that one. Here's how they start to mix things up. And again, this is not an overly challenging passage as a whole. It's not an overly challenging question, but this is an example of how the MCAT is going to test you. And this is where it gets fun, quite frankly. It says, if DNA ligase activity were selectively inhibited. Okay, now they talked about inhibition in the passage. And this is where I'm walking you through this, because that's the purpose of today. Right? The purpose of today isn't for me to give you a passage and questions and you to try to get them right? The purpose of today is for you to start to really understand how this thing is testing you. So they've just given you, I'm really kind of treating this like a forensic case, if you will. So if DNA ligase activity were selectively inhibited. Now, they're talking about DNA ligase activity being inhibited. In the passage, they talked about inhibition that they did, which was specific to helicase and topoisomerase. So they're already trying, right? And there's a reason behind this. They're trying to throw you off, because they're trying to get you to think, oh yeah, I totally saw inhibition. And that inhibition that they talked about in the passage, which I literally just read, was affecting the supercoiling, right? The tension, the torsional strain in the fork. But they're not asking you about the topoisomerase or the helicase. They're talking about, if we were to incorporate DNA ligase, or inhibit, DNA ligase activity, then what would the problem be? So immediately you should start thinking to yourself, and this is literally the process that you want to train yourself. This is how we talk about develop your critical thinking skills. This is the development of your critical thinking skill. Your critical thinking skill at this point should be, okay, DNA ligase is being inhibited. What does DNA ligase do? It's the glue. If I inhibit it, then the glue won't stick. True or false? If I took Elmer's glue or whatever brand of glue, I think Elmer's glue is still a thing, right? If I just took any type of glue and I did something to not make it sticky, if I inhibited its stickiness, then the glue wouldn't work, right? So then you have to ask yourself, well, where do we use ligase as far as DNA replication is concerned? And you go, well, they even told me in the passage that we use it to stick together the Okazaki fragments. So your logic is now going through, this is your critical thinking process. You're thinking, okay, my ligase isn't going to work. Therefore, I'm not going to be able to stick the Okazaki fragments together. Right? There's nothing about the Okazaki fragments not being made that's stated here. It's just I know that I'm not going to be able to stick them together. So I'm going to probably have a whole bunch of Okazaki fragments that are not going to be connected to each other. You always, I can't overemphasize this, you always want to and need to go into the answer choices looking for something versus hoping to find something, right? You need to be actively going into the answer choices saying, I want an answer that says something about the Okazaki fragments not being stuck together, or just Okazaki fragments that are not stuck together. You want to be looking for something. So looking at these four answer choices, which one equates to that? Which one aligns with that sentiment? Put it in the chat. Type in the letter. Tell me what the piece is that you see. B it is, right? B it is. So that's the one that you're going to want to choose, right? That's plain and simple, right? The accumulation of the unsealed Okazaki fragments, that's going to be the one, right? That's the one that's there. So it's, for some reason, didn't show that it's highlighted. There it is. Okay. So where'd you go? That is your correct answer, right? That is the one that is going to get you the points. Go through the other ones though, just for fun, right? Discontinuation of the leading strand synthesis. Ligase isn't going to have anything to do with the leading strand synthesis, right? Leading strand synthesis, that's continuous, doesn't require ligase, so we don't need to worry about that. Prevention of the helicase-mediated unwinding, that's independent of the ligase function, right? Ligase has nothing to do with helicase. And increased supercoiling ahead of the fork, again, that's topoisomerase, not ligase. So the only one that's going to work is going to be answer choice B. Right? This is, again, part of that thinking, right? That critical thinking, going through the process, as it were. Not too bad? Horrible? Where would you scale this one? Obviously, I've walked you through it. I've shown you kind of how to manage this thing, but not terrible, right? Not horribly terrible at all. Okay, not horribly terrible at all, for sure. Okay. Let's look at one last one. This one gets a little more kind of easing you into this, right? As it were, right? So we're like, okay, first one, very straightforward. Second one, straightforward with some, analysis that has to happen. Now, this is where, remember I was talking about the different skills I mentioned early on. There was the skill about really understanding, evaluating experimental design. This is one of those types of questions where they say, which of the following experimental designs would best test the hypothesis that topoisomerase is most critical during replication rather than during elongation. So this is where we go and say, okay, what did the passage actually tell me about, right? What did they, what was the information that was actually given to me? And more importantly, what's the series of events that's going to, kind of take things into account as far as the information that was presented to me in the passage itself, and then of course the information that I know, from before. So they're basically asking you to evaluate 4 different experimental designs, and very specifically, they tell you that the hypothesis is that topoisomerase is most critical during initiation, not during elongation. So when we think about topoisomerase, immediately, and type this into the chat, what is topoisomerase responsible for? I mean, you've heard me say it like 50 times already today, but say it one more time just for fun. What is topoisomerase responsible for? Everyone got silent all of a sudden. Maybe it's the delay. Topoisomerase, it's all about unwinding. That's right. Right, Tegan? It's all about unwinding, right? All about kind of relaxing things and making things, right? Relieving that supercoil tension that's ultimately there. So if we wanted to kind of evaluate things, we're probably going to be looking very specifically at the replication fork, right? That's chances are where we're going to want to be looking. So with this question, you do need to go through each of the answer choices, right? So compare the fork progression in wild type cells and in topoisomerase knockout cells. So wild type, of course, means it has everything. Topoisomerase knockout means you removed the topoisomerase over the first 5 minutes of the S phase. Now, here's my question. Why the S phase? What's so special about the S phase? Because we haven't talked about the S phase. The passage didn't mention the S phase. What's so special about the S phase? What happens during the S phase in the life cycle of a cell? DNA is synthesized, right? That is the synthesis phase. That's why it's called the S phase, right? So that's where you actually have the DNA replication that's occurring. So we wouldn't want to be looking at the fork progression during, during the mitotic phase, right? That doesn't make sense. You want to look at it during the growth phase because there is no fork progression at that point. So S phase makes sense. If we look at it during the first 5 minutes of the S phase, that's when things are getting started. So that would make sense as far as the fork coming into play because that's where your DNA replication is happening. Good. So we're going to hold on to that. Measure the nucleotide incorporation rates in cells treated with helicase inhibitor after initiation is complete. So helicase inhibition confounds the specific role of topoisomerase, but it's not the thing that you would want to be testing to prove this hypothesis that topoisomerase is the most critical during replication initiation rather than in the elongation. Because now we're talking about the actual incorporation rates, right? The actual bringing in of the nucleotides as they're coming through. And we saw from the passage that, they're the same in either direction overall. And we saw from the passage as well, right? They told us that we had this, 60% drop off, right? So it was when the cells were treated with topoisomerase inhibitor, the fork progression went down by 60, but adding the inhibitor after unwinding had already begun. That only produced a 15% decrease. But that was specific to topoisomerase. This case here, we're looking at, this is about helicase, so that's not going to be helping us, as it were. You look at C, and it says add topoisomerase inhibitor after 50% of the genome has replicated and measure the Okazaki fragment length. If you are going to be adding an inhibitor later, after 50% of the genome has been replicated, are you in fact testing initiation or elongation? What would you be testing if you're adding the inhibitor at the 50% mark, once you've done 50% of the genome? All of it is replication, but is it initiation or is it elongation that you're testing if you're adding at the 50% point, the halfway point? It's elongation. And the question very specifically was asking you to be testing the replication during initiation, right? During replication initiation. So here, the problem with the answer choice is that it's the wrong period of time, right? It's the equivalent of, something happened in the 1950s, but you're talking about the 1970s, right? You you're in the wrong year, right? You're in the wrong place, as it were. Answer choice D says to quantify the SSB binding in untreated cells lacking DNA ligase. Problem here is there any connection between the SSB binding and ligase and topoisomerase? Are they related at all? Yes, no, right? There's no connection, right? There's no connection between the two. And so as a result of that, you can eliminate answer choice D as well. But answer choice A is perfect, right? If you say, okay, I'm going to compare that fork progression during the first 5 minutes of the S phase, I'm going to be able to tell you whether or not that topoisomerase is going to be critical or not, because I've got these two groups, right? I've got group 1, group 2. Group 1's got everything. Group 2 has no topoisomerase. Let me see after 5 minutes where we are as far as those replication forks. Let me draw a comparison between the two. If I design that experiment, regardless of what the outcome is, that's the experiment that's going to tell me whether or not this topoisomerase is critical during the replication rather than during the elongation, right? During the initiation versus the elongation. Too many IONs is too many shuns in the question. So, okay. So that's how you have to think about these things. You have to think, okay, let me go through this step by step, process by process, to see kind of where things fall into and where it all comes about. So these were 3 very representative questions, not just for the biochem section, but really for all of these, as it were. Victoria, it's not a short question, and it's not short answer choices, which actually brings up just a thought that comes to mind. When you're taking this exam, you're going to need to do a little triaging, right? You're going to need to sometimes say, you know what? This is a question I'm going to answer right now. This is a question I'm going to answer later, right? That's okay, right? That's totally okay. You can do that, and you need to be comfortable doing that. You need to be comfortable saying, I'm going to make some choices, and right now, this question, I can probably do it, but it's going to consume a lot of time, and I want to go and get some other questions right, or vice versa, right? That is all very, very doable as you're going through. So something to bear in mind for certain. A little bit of news. I'm sure you've seen it at this point. Nothing too new. But there was a big announcement that came out recently. It wasn't about a pop star and a football player. It was about the MCAT registration dates and the MCAT test dates for 2026. Registration for the MCAT will open on October 21st for those of you that are on the eastern side of the United States, October 22nd for those on the West Coast. The test dates have been released for 2026, so it's not too early to at least start looking at when you're planning on testing so that you can start to plan out your preparation. we're going to be hosting an event, not next week, but the week after that. So next week, we're going to do another one of these, but it's going to be about psych-soc. The week after that, we're going to do an event specifically about like picking a test date. We have some articles that are going to be coming out very, very soon. So keep an eye out for that through our pages. But bottom line is start thinking about what your plan is for testing in 2026. If you're testing in 2025, meaning you testing in the next couple of weeks, essentially, great. But if you're testing in 2026, now is the time to start at least considering like when you want to test, what other things you're going to have going on in your life, school, work, research, all the things. Make sure that you kind of plan things out as it were. Janie, good question. Good strategies in the past seem long and overwhelming, especially with figures. If they're long and overwhelming, that's usually an indication that there is a lot of detail and a lot of extra information that's in there. And so the strategy, let's assume that we're not talking about timing, right? We're just talking, you're going to do the passage. The strategy there really comes down to, as you're reading it, to not get bogged down in the details and to not get bogged down in the figures. The biggest mistake that I've seen students make through the years when it comes to passages that are just full of information is feeling the need to do a full analysis of the figure, a full analysis of the table. If they give you, they do the experiment, they've got 4 or 5 different experimental results, or 4 or 5 different trials with all the different results, feeling the need to go through and do a full analysis the same way you would do an analysis for a lab report or for a paper that you might be publishing. Don't do that. Go to the questions and let the questions dictate to you what analysis it is that you're going to need. Let the questions dictate to you what details it is that you're going to need. Take note of where information was mentioned, but you don't have to remember what the details were, right? In paragraph 2, they told me how much of X they added to Y. How much of X they added and how much of Y they had, that's in the passage if you need it, right? So that's the strategy in terms of those passages that are kind of overwhelming. So hopefully that helps. But that's where I would say to kind of begin with all these, as it were. We do offer full MCAT test prep tutoring services. So kind of all our hallmark is that everything is very, very bespoke and tailored around the student. We've got a phenomenal team of faculty that we have on board. All great test takers, all great scorers as far as the MCAT, but more importantly, individuals who are very, very invested in getting you comfortable with this exam and really being your coach and mentor and guide through this material and breaking down the complex concepts into really what the test is ultimately testing you on at the end of the day. So if you're interested in finding out more about that, just want to kind of chat about the different options that are available to you as you're figuring out your timeline, grab this QR code, set up it's a free conversation with me. And we can talk about kind of what would work for you as far as timeline is concerned and when to get started with your prep and all those things as it were. So keep that in mind as it goes. If you've got questions, now's the time to ask them because we've got about 5 minutes left. I know you've been asking questions as we've been going. I think I've answered all of them at this point, but if you've got any more questions about anything related to this, hopefully you had some fun. Hopefully you learned something, got a little bit of exposure to kind of how this test or how this section works. Come back next week. We'll have Psych-Soc on the docket. I'll be honest, not as exciting. The Psych-Soc section is, I think, still in its evolution, as it were, as a test section. But it's good for you, is what it comes down to. But if you have questions about anything that we talked about here today, feel free to chime in with them. So Shruti, good. That's a great question, actually. Let me go back to. I want to do some maths with you. Is what I want to do. I gotta. This thing doesn't let me jump back to a slide. Let me just go to a number. Unfortunately, I have to go through them each. It's one kind of pet peeve of mine that I don't like. I'm like, I want to just be able to jump to a slide, but give me a second here. We're getting there. We're getting there. We're getting there. There we are. So generally good kind of math, if you will, is give yourself a minute per discrete question, right? Those are the standalone questions that are on the exam. You've got 15 of them, right? So you say, okay, I really have 80 minutes or so for all the passages. You're going to have between 9 and 10 passages. So you're looking at roughly 9 minutes or so for the passages as far as the test is concerned. So that's the way to think about it. That being said, there are going to be some passages that you're going to need and want to spend more time on. And there are going to be some that you're going to spend, or you can spend considerably less time on. So it's an average is what it is, right? It's kind of a ballpark, if you will. Sometimes there's, more questions for one passage versus others. So obviously that's going to take more time. So, and when I say that number, I mean the questions and the actual passage. If you're asking about the time to read the passage, I would target somewhere in the neighborhood of 4 minutes or so to read it, map it, extract the information, and then get to the questions. The questions are where the points come, right? So you need to get to the questions, and you can always go back to the passage. What you don't want to be doing, and it gives me a headache anytime somebody says this, what you don't want to be doing is reading the questions first and then going to the passage, because then you have to go back and read the questions again. Then you're just wasting time. So don't do that, in my humble opinion. But Richard, I'm glad I made you feel better about the MCAT. You're very welcome. You're all very welcome. Vuk, you're very welcome as well. So I'm glad it was helpful. Any other questions about all the things? Shruti, absolutely. So good question. While you're doing content review, should you prioritize the high yield topics? Absolutely. That's where more points are, right? It's a balancing act, right? So definitely the higher yield topics. I always tell this story, so if you've heard it, I apologize, but I love the story because it's just so perfect. I had a student who literally a couple days before the MCAT freaked out about alpha and beta decay because she'd seen one question and had gotten it wrong. And I was like, don't worry about it. She's like, no, I got to study alpha and beta decay. I was like, don't worry about it. Super low yield topic. Sure enough, she didn't get all any questions about alpha and beta decay on the actual exam. So yeah, high yield always first. Always first. But don't ignore the other stuff. But prioritize it for sure. Like thermodynamics, 100% will be tested on the MCAT. I can guarantee you that. If you don't see a single thermodynamics question, then I will find a hat and I will eat it. So hopefully nobody comes to me and says, with that said, thank you all very much for joining us tonight. Stay tuned. Lots more great stuff coming from med school headquarters, from our team of phenomenal faculty over the course of the next year. You're very welcome, Omid. And seeing that you said another great session, I assume you've been to others. So thanks for tuning in is what it is. Let us know if you have any questions. Feel free to reach out on social, all the places, and we'll be more than happy to help you out. Come to MapCon and come next week. We'll finish out Psych Soc, and then we'll do something else. So have a great night, everyone. You're all very welcome. Take care.